Okay, so we're going to pick up where we left off talking about the different organelles in the cell. All right, so first we're going to talk about the endoplasmic reticulum. Frequently that's abbreviated ER, all right, endoplasmic reticulum. And so the endoplasmic reticulum is this kind of network of membranous channels. Okay, so in this picture you can see we have this kind of network, whoops, let's try the laser pointer, there we go, this kind of network of membranous channels here and this other network. A lot of times, right, these are actually going to be connected to each other, right, so they would be interacting with each other, right, but there's these membranes and you have these little bits of membrane that'll kind of pinch off and move from here to the next one, right, or from here to another organelle, okay? So that's the endoplasmic reticulum, the ER. There are two types of ER. There's smooth ER and there's rough ER. So the rough ER is going to have ribosomes attached to it and the smooth ER will not. So let's talk about smooth ER first. Okay, so smooth ER has no ribosomes. All right, and then let's talk about the function of smooth ER. All right, so if we were to point at it, that would be this one here. So it doesn't have ribosomes, which are those little purple dots. So that would be our smooth ER. You can also see that it's connected to, both endoplasmic reticulum are, they're connected to the nucleus. So they're actually going to be continuous with that outer membrane of the nucleus. Okay, so it would actually look like that if you could see the outline for it. All right, so they're continuous with the nucleus. And so the function of the smooth ER, it has many functions. So first of all, it functions in detoxification. And so this would be of different drugs and alcohol and that kind of thing. All right, so the smooth ER would help to detoxify those substances. So an example of a cell that would have a lot of smooth ER in it would be liver cells, because liver cells function in detoxifying, so they would need a lot of smooth ER. So detoxification. Also carbohydrate metabolism, right? Breaking down carbohydrates is going to happen in the smooth ER. Also calcium storage, okay? So the smooth ER, and I'm going to kind of just move this out here so we have more room. In some cells, like muscle cells, can store calcium, okay? And we're going to see this when we talk about muscle contraction. So this would be in muscle cells. Okay, they're going to store calcium and then they can release calcium when the muscle is going to contract, and that's going to play an important role in muscle contraction. Right, and then one last function of that smooth ER is steroid, fat, and phospholipid production. All right, phospholipid synthesis. So synthesis meaning we're making something. Okay, so we're going to make steroid fats and those hormones, as well as including cholesterol, as well as phospholipids in the smooth ER. Okay, so that's 4 different functions for smooth ER: detoxification, carbohydrate metabolism, calcium storage in some cells like muscle cells, and phospholipid or steroid fat synthesis. All right, so now let's talk about the rough ER. Okay, so rough ER is different because it has ribosomes attached to it. All right, so this would be our rough ER here with its ribosomes attached. Okay, so let's talk functions of that rough ER. All right, so functions. Okay, and so the rough ER, the function is pretty straightforward since it has ribosomes attached, right? The job of those ribosomes is going to be to make proteins, and that's actually the function of that rough ER, is to make proteins. Okay, and so cells that would need a lot of proteins made are going to have a lot of rough ER. All right, in particular, neurons would have a lot of rough ER. Muscle would have a lot of rough ER, all right, to make the proteins for muscle contraction. Okay, and so these proteins are going to be either membrane proteins, which we talked about before, right, or they're going to be proteins that are sent out of the cell. They're exported from the cell. Okay, so exported proteins. Okay, so that's the rough ER. All right. So now let's talk about the Golgi apparatus, because its function is kind of closely associated with the endoplasmic reticulum. Okay, and so the Golgi apparatus is also this series of membranous sacs, right? So this is going to be our red organelle here. And so we have all of these kind of red curved membranous sacs, all right, that kind of stack on top of each other. And the function of the Golgi is to package secretions with things like proteins for export, all right? So it's kind of the UPS center of the cell. All right, so package, secretions, things that are going to be secreted, right, released from the cell for export. And so an example of this would be a protein, all right, made in the rough ER, right? So protein from the rough ER. Okay, so let's talk about for a second what would happen, right? Maybe let's just follow kind of a protein from where it starts to where it ends. Okay, so if we have a protein that's going to be made in the rough ER, right, first we need to make a copy of that DNA, right? So we'd copy the DNA into a little piece of mRNA. That mRNA would travel out of the nuclear pore, all right? It would go and it would find a ribosome. So let's say we found this ribosome here, right, to make our protein. That mRNA would give its information to that ribosome. That ribosome, I'm going to zoom in a little so we have a little more space. That ribosome would make that protein, and that protein would form on the inside of our rough ER. Okay, so then from there, that rough ER would package it up and send it out. All right, so it would move it over to one of these little bubbles. All right, and then that bubble would pinch off and head over to the Golgi. Okay, so it would merge with the Golgi, and that Golgi apparatus would now be modifying that protein. All right, as it moves from one kind of layer of the Golgi to the next, it's going to be modified slightly, and then it is going to reach our kind of end point here. Okay, and so these pieces that are moving off of the Golgi, right, these little bubbles, they are called vesicles. Okay, so vesicles, there are two types of vesicles that can come off of the Golgi. You can have a transport vesicle, and so a transport vesicle is going to go between compartments within the cell, right? So this is going to be between compartments or organelles in the cell. Okay, so if I were to point to a transport vesicle here, right, it would be, and I gotta draw a long arrow, right, that would be this guy here. okay, because it's going from the Golgi, right? We have our transport vesicle, and it's making its way over here to this other organelle we're going to talk about called the lysosome. Okay, so it's just going from the Golgi to the lysosome, so it's staying inside of the cell. All right, so that's going to be different from our secretory vesicle, which sends things out of the cell, right? So it exports out of the cell. Okay, and so that would be, I'm going to crisscross these lines here, that would be this guy here. Okay, so it's going from the Golgi to the cell membrane, and then whatever its contents are that it has in here, it will release outside of the cell when it reaches the membrane. Okay, so secretory because it's secreting something out of the cell. All right. Okay, so that's the Golgi. It kind of packages things up, sends them off to where they're supposed to go, gives them kind of their shipping label and saying where they're supposed to go, whether it's to a different organelle, right, like the lysosome, or maybe to the mitochondria, or maybe back to the endoplasmic reticulum, or this secretory vesicle, which will send it out of the cell. Okay, so now let's talk about our lysosomes. Okay, and so our lysosomes, which we have here, I've drawn it in yellow. Okay, so our lysosomes are going to be organelles, right? They're membrane bound, so they're membranous organelles with a lot of digestive enzymes inside. All right. And so those digestive enzymes, there are many different enzymes, right, that can digest many different things. And so the contents of that lysosome could be very dangerous to the cell if they somehow escaped, right? So if this lysosome somehow, burst, right, and let out its contents, those contents could potentially harm the inside of the cell, start digesting stuff, other organelles and things like that, okay? And so there are two things that help to protect against that. And so the first thing that protects against that is the fact that this is actually a membranous organelle. So it has a membrane around it, and that membrane keeps its contents, right, the inside, separate from the rest of the cell. Okay, so that's one barrier. The second kind of thing that's protecting the cell from all those enzymes is the inside of a lysosome has a very low pH. Okay, so it's very acidic on the inside, and that is because those enzymes have to have that low pH in order to function. Okay, so this low pH is required for those enzymes to function. Right, so enzyme function. So that kind of protects the cell also, because if for some reason this lysosome did burst, those digestive enzymes would get out here in the cell where the pH is much higher and they wouldn't function. Okay, so it's kind of like a safeguard. All right, so that protects the cell from those digestive enzymes. So let's talk about the digestive enzymes briefly and what they do. And so the name of an enzyme a lot of times will tell you its function. Remember, all enzymes end in ACE, so you can see all of these have ACE at the end. All right, so let's talk about what they're going to be digesting. So a polysaccharidase would digest a polysaccharide. All right, so these would be digesting things like starch and glycogen and that kind of thing. Okay. And then proteases are going to digest proteins. All right, so proteases digest protein. Lipases digest lipids, so fats. Nucleases digest nucleic acids, so DNA and RNA. All right, so different enzymes that are going to digest each type of organic molecule that we talked about in our last section. Okay? All right, so those are our lysosomes. Okay, and so then there are also peroxisomes. All right, and I don't have any peroxisomes drawn on this picture here, so we'll just talk about the function of a peroxisome. All right, so peroxisomes are membranous. Okay, so they're surrounded by their own separate membrane, and they function to detoxify. All right, so similar to the smoothie R, right? Peroxisomes are going to detoxify things. They're going to detoxify things by oxidizing them. All right, so they detoxify, right, by oxidizing. Okay, so they're going to detoxify drugs and other things like that, similar to the smoothie R, through oxidizing. Okay, they're also going to function in breaking down, right? Metabolizing. And you'll learn about this with the digestive system, fats. Okay, so if your body needs to burn fat for energy, that fat is going to be broken down by a peroxisome, right? So for ATP, so they're involved in that process. Okay, good. So that's our peroxisome. All right, and so then our last topic, we have centrosomes. All right, so our next to the last topic is the cytoskeleton. And so the cytoskeleton is not drawn in this picture because essentially it would fill this entire picture with a bunch of kind of crisscrossing lines, right? And that would be really confusing. Okay, so the cytoskeleton is the actual skeleton inside of the cell. And so you need to know two components, right, that make up the cytoskeleton. The first one is microtubules. All right, so microtubules are going to be hollow proteins. Okay, and so they're these kind of hollow protein tubes. All right, so they're these kind of like columns, these tubes of protein found in the cell. And that protein that they're made of is called tubulin. All right, so microtubules are made of a protein called tubulin, and they form these hollow tubes, right? That's a lot of tube-related words. Okay, and so whenever you see microtubules, you should automatically think of movement. All right, so if there's microtubules involved, there's going to be some sort of movement involved. Okay, so let's talk about some functions of microtubules, some specific examples. All right, so one example of a microtubule is going to be in a flagellum. All right, so you see flagellum, the only cells in the human body that have a flagellum is the sperm. Okay, and so that is that tail on a sperm that allows the sperm to swim. That tail is filled with microtubules, and that is what gives it its ability to move and kind of whip back and forth. Okay, and then some other cells, like in your respiratory tract, are going to have structures called cilia. All right, so cilia are going to be like these little hairs on the surface of cells, and they actually will move, right? They'll beat and kind of create like this current, and actually that's what helps to move mucus in your respiratory tract, up and back and down your respiratory passage. That's actually why when you get like a cold, your nose starts running. That's because when you get a cold, these cilia become very lethargic and they're not functioning quite as well. And so I'm having allergy issues, right? So I'm sniffling a little too. All right, and so those cilia, because they aren't beating as well, right, moving, that mucus is not being moved, and so then, your nose will start running and that kind of thing. Okay, and so that movement in the cilia is provided by microtubules. All right, and then another function we're going to talk about at the end of this chapter is mitotic spindles. All right, and so in mitosis, these mitotic spindles, right, these threads of microtubule form, grab a hold of the DNA and pull the DNA to either side of the cell, right? So we're moving that DNA to either side of the cell. So movement again with our microtubules. Okay, good. So now microfilaments, right, are another type of cytoskeletal element, right, or kind of little fiber, right? They're kind of thread shaped, both of these. And microfilaments are made of a protein called actin. Whoops. All right, so the protein that makes up microfilaments is going to be actin. Okay, and microfilaments give the cell its structure. And we're also going to see actin when we talk about muscle contraction. All right, so structure and shape to the cell. Okay. All right, and then our last organelle is going to be the centrosome. Okay, and the centrosome is another important organelle for mitosis, right, that we're going to talk about in just a second. And so the centrosome is going to contain 2 centrioles. All right, so the centrioles, they're typically going to be kind of like these little rod-shaped structures, right, like this. All right, and they're going to be at a right angle to each other like this, right? So they kind of sit next to each other. There's these little rods. And then the area that they're found in, right, so this whole kind of area they're located in is called the centrosome. So the centrosome contains 2 centrioles, okay, and the function of those centrioles is to make those mitotic spindles. They make microtubules. All right, so they're going to make microtubules. All right, so specifically those are those mitotic spindles. So they'll be really important during mitosis. All right, so also this centrosome, right, it's just kind of an area, so it is non-membranous. Okay. All right, so those are all of our organelles inside of our cell. So before we talk about cell division, we're going to spend a little time talking about cell transport. All right, so with cell transport, there are 2 types of cell transport. So it can be either passive or it can be active. And so with active transport, it is going to require, all right, so it requires ATP for active transport. Passive transport does not require ATP. All right, so it does not require ATP. Okay, and so the reasons for that is that passive transport, molecules are going to move along their concentration gradient. All right, so they're moving from high to low concentration. All right, so this is from high to low. Okay, and that doesn't require energy, right? That's like water going over a waterfall, right? You don't have to use any energy for that. Okay, so it's going along its concentration gradient from high concentration to where there's a lower concentration. And then active transport is typically going to be against the concentration gradient. All right, so you're going to be going from an area of low concentration, all right, to an area of high concentration. Okay, so that would be, like pumping water, right? So maybe your basement floods, right? And you have all this standing water, you want to get it out. and you're going to actually pump it, right? So you need to turn on that pump to suck up that water and move it out, okay? And so you're going to go from an area of low concentration to high concentration, right? Or a lot of times in the cell, right, you're going to be taking a molecule. So for example, we said before, if we have the cell membrane here, we have a ton of sodium on the outside, right? We have a tiny little bit of sodium on the inside. And so constantly the cell is going to be pumping this sodium out, okay? And that is going to require ATP because you're going from where there's only a little and you're cramming that sodium out to where it's already really crowded, okay? And that requires ATP. That's going to be active transport, okay? All right, and so in general, with the cell, right? Remember, the membrane is made of a phospholipid bilayer. And so in order for things to move through the membrane, right, those things have to be, so without any kind of aid, those things have to be nonpolar, right? Like that lipid of the phospholipid bilayer. They would have to be very, very small. And so if they're not nonpolar or very small, all those other things would need some sort of carrier protein to help them through, okay? So what molecules need transport, all right? Those are going to be polar molecules, molecules that are large, all right? And then also molecules moving against that concentration gradient, right, are going to need some help in order to be able to move against that concentration gradient, okay? All right, and so we say the membrane is selectively permeable, so we just mentioned that the membrane allows nonpolar and very small molecules through. All right, so most of our polar kind of organic molecules are going to be too large to move through the membrane without some sort of help. Okay, so they have to be either very, very small or nonpolar to just go through the membrane without any help. Okay, so let's talk about first passive transport. All right, so remember, passive transport does not require ATP. And we're going to be going from high concentration to low concentration, all right? And so our first type of passive transport is the simplest. So this is diffusion. And so with diffusion, substances are going to move from high to low concentration. All right, so it's going from high to low concentration. All right, and so think about, we had our waterfall example. All right, so another good example would be if you have like a water balloon, right? You poke a hole in that water balloon, that water is automatically going to start leaving that water balloon because it's going from where there's a lot of water, right, on the inside of the balloon to where there's less on the outside, right? That is going to be moving along its concentration gradient. So from high concentration inside of the water balloon to low outside. All right? And so simple diffusion like this would be for small and nonpolar molecules. So these would be molecules that could just move across the membrane without any help from high concentration to low concentration. All right? So the only thing that determines if they're moving through the membrane or not is that concentration gradient. All right? So then another kind of form of diffusion would be facilitated diffusion. So facilitated diffusion would be for things that require help to get through the membrane. So these would be for large and polar molecules. Okay, and so they're going to need some sort of assistance. All right, and so these involve transport proteins. All right, and we talked about membrane proteins before, and so the transport protein that would be involved here for facilitated diffusion would be channel proteins, right? Remember, they just form like a tube through the membrane, allowing things to move as they want to through, right? So things through that channel could go from high concentration to low concentration just through basically an open tube in the membrane, all right? And so some examples of this would be ions, right? So definitely, and we're going to talk about this with the nervous system and with muscle contraction, ions, right, like sodium and potassium are going to have these channels, right, that will allow them to diffuse through the membrane. So it's diffusion, but it's facilitated because it needs this transport protein to actually get through the membrane. This diffusion that's not facilitated would only be for things like nonpolar and small molecules that don't need a transport protein to get through. Okay. All right, so our last topic for this video is going to be osmosis. So our last kind of specific type of passive transport. So osmosis specifically involves the movement of water. Okay, and so water, because it is passive transport, is going to be moving from high to low concentration. I always find that a little harder to visualize with osmosis, and so I prefer to say that water moves always, right? Water moves towards more salt, right? So more salt or solute, really. Our examples are going to be of salts typically, but it could be towards glucose or some kind of dissolved solute. Okay, so water loves solutes. So wherever there's more of a solute, water is going to go there to try to dilute that solute out, right? Okay, so let's look at some examples. Oh, before we do, all right, so water also, typically its movement is going to be facilitated, okay? And so that means it has a protein. And the protein for water, it has a special name. So water moves through what's called an aquaporin. All right, so an aquaporin is a channel for water. I think that's a really cool name, an aquaporin, right? So a water pore. Okay, so now let's talk about the movement of water and kind of the different concentrations of salts that can cause water to move. Okay, so if we go with our cells here, okay, so we have cells under 3 different conditions drawn here. All right, so our first condition, and one thing to remember with cells is that they always have a salt concentration in their cytoplasm, right, in that cytosol of around 0.9%. All right, so all of these guys on the inside would have 0.9%. 0.9%, and that's for salts. Okay, cells also have a glucose concentration, and this is important if you're going into the medical field, of about 5% on the inside, right? So salt is always going to be 0.9%, and glucose is going to be 5%. This is always going to be what it is on the inside of the cell. Okay, so I would commit those values to memory. All right. So a cell, if you take a cell, drop it into, a water solution with salts in it, all right, you should know what's going to happen to that cell in those different solutions. Okay, so if we take a cell and we drop it into a solution that has salt solution of 0.9%, right, just like the cell itself, okay, so far as water is concerned, right, there is no net movement of water. Okay, so that water is not rushing out of the cell, water is not rushing into the cell. There's no real net movement of water. Okay, and that's why most IV solutions, right, just as a simple IV solution, like an IV saline, is going to have a salt solution of 0.9% and a glucose of 5%, right? That's a typical IV saline solution, okay? Because you don't want to give IV to a patient and cause, one of these reactions in their cells. Either they'll burst or they'll shrink up, and possibly die, okay? All right. So that's isotonic. So isotonic means the solution has the same salt or solute concentration as the inside of the cell. All right, that's isotonic. Okay, and so then for a hypotonic situation, you would take the cell, all right, and you would place it in a solution that has a much lower concentration of salt. And so remember, our rule is that water always moves towards more salts or more solutes. Okay, so water is going to rush in towards that 0.9%, which is much more than 0.01%. All right, so H2O, water, is going to move in. All right, so it's going to move into the cell, and that is going to cause the cell to swell, all right, and the cell will eventually burst. That is called lysis, or if we're talking about an IV solution, so we're talking about blood, right, hemolysis would be when a red blood cell bursts. And so that can happen if you give a patient a solution that doesn't have enough salt in it, right? So that can be very bad. Okay, and then our last possible solution is called a hypertonic solution. And so you can probably guess this one has a higher concentration of salt. So if we put it in, let's say, something extreme, like a 10% salt solution, water always moving towards more salts, right? That water is going to move away from that 0.9% towards that big fat 10% of salts to try to dilute it. Okay, so water moves out. All right, so that cell is going to shrink up because it's losing water. And so we call this shrinking of the cell crenation. All right, so crenation is the cell shrinking because it's losing water. Okay, and so you should understand each of these solutions and the effect they have on the cell and why. So which way is water moving in response to that solution? All right, and so then on the next video, we'll pick up on active transport.